I'm here coming from the IDIAP Research Institute, and I'm not sure if everybody has heard of this institute, so I have one or two slides just to introduce it before I will start with my scientific presentation. The first point to mention is that IDIAP is in the Valais region of Switzerland, so that there is an illustration of the Valais region. It's a narrow strip in between two regions of the Alps in Switzerland. And the institute itself is a research institute entirely focused on AI. It's around 35 years old. In fact, I think we celebrate 35 years this year. And it covers the full breadth of AI research from fundamental to applied. My group is focused on AI for health, more on the applied side, and with a focus specifically on human-centered research, which includes this topic of knowledge alignment, which I will come back to. But that's the institute there, and that's the view out of my office window. So do think of paying us a visit in the future. So as we all know, generative AI is rapidly, as was mentioned also in the introduction, it's rapidly transforming the way that research and practice are done throughout biochemistry and health. One of the most significant breakthroughs in recent years in this area was, of course, AlphaFold, and that was recognized with the award of some Nobel Prizes, which you can argue are a flawed institution, but nevertheless, it counts for something, back in 2024. And this was for this model, which is trained from basically protein sequences and from homology, and incorporates also a whole lot of prior knowledge about protein folding constraints in order to be able to predict protein structure from measured sequences, which had been a problem for 30 years or more. And this is not at all restricted to protein folding. The sort of impact of generative AI throughout health and medicine is broad. There have been many summaries of the different ways that this is impacting research and practice. And this particular one is just one of them, which I like because I like this figure, which sort of brings together a lot of different things. On the one hand, it mentions research and the different ways that research is being transformed. And this is everything from supporting experiments to really these foundation models, which we will probably be mostly focusing on today, to sort of structuring data and extracting more value out of the data that we do collect, which is really unlocking more possibilities for research. But it's also transforming patient care. I know that many aspects of patient care, whether explicitly or implicitly, are being transformed by the presence of generative AI. What I mean by explicitly and implicitly there is that patients themselves are now consulting large language models for their own health advice, and many, many doctors are consulting large language models for support in various ways. And this is even though these models have not been approved for those applications. On the other hand, there are approved uses such as supporting medical documentation, and this is rolling out throughout hospitals across the world. And on the other hand, we see transformation also in education, and here there's a bit of a push and a pull because it may be more difficult to educate certain knowledge-rich fields, to educate students who have access to this kind of technology. And in the olden days, you would sort of send students home to write an essay. But these days, at least my experience teaching at the university, is that if you send students home to write an essay, you get back something that was generated by AI. And you have to ask yourself, well, what's the point in grading that? What's the point in producing it? But this is part of our education. And on the other hand, there are many valuable uses of the technology for supporting the learning process. And we have to navigate the sort of tension between positive and negative transformation in that space as educators. So a very broad and diverse landscape today. Oh, that was slightly too quick. Today I will focus on my group's research in the field of biological AI, I guess, or biochemical AI. And here as well, there are many exciting many exciting research projects are going on, and I just want to highlight a vision which is presented in this Review and Perspective article, which is this idea of generalist biological artificial intelligence, or modeling the language of life. I think this is a grand expression, but what it means in practice is this idea that you have this sort of, by training models with all the information and data that we can measure, and figuring out how to align those models across the different layers of things that we can measure, we have the possibility of sort of doing more from what we can measure than what we could ever have hoped to do when we could only use those different data sets in isolation. So building composite and interlinked maps of these different aspects of biology, from the DNA sequence to the RNA sequence, proteins, and everything else that happens afterwards, all the way up to the complex cellular context. And this is a great vision, but this is not in practice very easy or possible to do, because each of the different layers have very different measurement and informational kind of content, and different sort of strengths and weaknesses of the signals that they encode, and different sources of noise and sources of variability. And what tends to happen in practice when you put together multiple layers of these different channels is that one will drown the other out, or it's very difficult to find the right balance to sort of have signals coming through from different channels at the right strength. And some have very, very little data, while others have lots and lots and lots of data, but that data is not encoding a huge amount of information that we can use, and so on and so forth. So this vision, which is a wonderful one, I think, is still in practice a work in progress, which leads to many, many research questions, which is why we all still have jobs. Here's just another figure from that review article, sort of delineating the different aspects where this vision is anticipated or is already having an impact, including scientific discovery, the generation of new molecules. This is an active area of research, sort of synthetic biology, as well as deriving new knowledge and new hypotheses from the existing measurements that we have. So my group works a lot on chemical language models. We like small molecules, partly because these models are smaller, and also the molecules are very information rich. And you see here a sort of example of a chemical language model. And chemicals are very interesting because you can represent these molecules very neatly in a sort of linear representation, which is well suited to sequence-based models, but they also can intrinsically be represented as a graph, and this kind of chemical graph representation is well suited to other sorts of foundation models or architectures. And there are many different strategies to doing sort of training or pre-training, unsupervised training of these models, including the classical masked sort of prediction. So you mask a bit of the input sequence and the model has to learn to predict it back. And we have relatively widely accessible sources of training data for these models, such as PubChem, which is a source of about 100 million distinct molecules, which can be used for this pre-training. And with that amount of data, you come up with a model which is relatively good at understanding and predicting sequences of small molecules. Different architectures offer complementary strengths and weaknesses. So you have in this space, you can use, and we do use, transformer-based models, which work from the sequences. And here you use a kind of linear representation in order to train the model. And transformer-based architectures are very good with long-range dependencies. So they can learn complex and subtle relationships between quite widely separated parts of the input molecule. This is driving their strength also in natural language processing. But on the other hand, you can use graph-based, so raw graph-based graph neural networks. And these are better at learning local patterns, such as complex ring structures and the differences between different complex ring structures. And neither in our hands, the graph neural networks are slightly better overall, but not for all classes of problem, not for all classes of molecule. And in fact, it's very interesting for us to study the sort of properties of problems that are better addressed in this space with sequence-based models, or those that are better addressed with graph-based models, and try to understand what this means for our future. Okay, another area where we work in, and I mentioned this specifically because of the problems that it gives rise to, is RNA language models. So proteins are a quite well-populated space. Lots of people work on proteins, great work going on. And for that reason, it's maybe sort of difficult area to really make a huge breakthrough in, but there are not as many groups working on RNA, and one of the reasons for this is that there's simply much less data available in RNA, so there are far fewer data in public databases for raw sort of RNA sequences, and also the problems are much harder on a biological level. So these molecules are transient, they're serving functions which are very context dependent within the cellular context. So the same exact molecular sequence could have a totally different function in another context. And we don't, or haven't sort of got the data which can really, with those annotations, which can let us break this all down and figure this all out and represent all of that. So it's a hard problem to predict what RNA sequences are going to be doing in a particular context, in a particular setting. And we want to improve this by figuring out how we can use more of the prior knowledge that's available to us in more sophisticated ways in order to make up for this sort of lack of basic brute force power of huge quantities of data that's available. Okay, so there are well-known challenges with this vision of biological AI being sort of solved by generative AI and language models. And one of those, as I mentioned, is this need to learn from very heterogeneous and small data sets, different sources of variability, different sources of noise, and to somehow detect the signal and abstract away from the noise. There is also still a challenge with bias in the data that is available for training, and a need to, and a challenge with generalizing far beyond the training distribution. This is a problem even for AlphaFold, although the training distribution for AlphaFold is quite representative of the majority of the problems that we could be interested in within a typical or traditional biological space. But nevertheless, if you apply AlphaFold on a sequence which is not homologous to any that are in the training data and is very far removed, it doesn't perform that well. Complexity and context dependence. This is what I mentioned about the fact that the same sequence or the same molecules could have very different behaviors in different contexts, and the data simply doesn't have that context built into the data, so we can't learn it. And this is something which we have some very small data sets which do have that context built in, but we need to generalize way beyond these small data sets. And then there are problems with consistency and reproducibility, and a problem of trust in general, but maybe more particularly a sort of challenge where we want to use these models to go beyond what we know, but then how do we verify that they're doing something useful if we are beyond what we know, right? So we need to figure out ways to build in useful feedback loops which help us to sort of ground the models and apply them consistently. And interpretability is a part of that process. We have to be able to figure out if the model is doing the right thing in this area of the unknown, in this frontier of the unknown biology that we want to go into. And in order to verify if the model is doing the right thing there, we need to have a way to extract something that we can test. So the model needs to be able to give us something that we can connect to one of our theoretical frameworks. So this motivates the sort of need to look at prior knowledge in biochemistry and health, to keep looking at it, even though we have these super powerful, performant, large-scale data-driven models. So that was useful. Thank you. I think I might take a second. So there are many different types of prior knowledge which we have been used to working with in biochemistry and health. Mathematical models encode knowledge in a certain way, and what we can call semantic or symbolic models encode a different type of knowledge in a different way. I mean, underneath all of knowledge representation is some sort of mathematical formalism, but the semantic and symbolic models have a commitment to also having a kind of meaningful layer, meaningfulness layer, which is different to the sort of purely mathematical or constraint-based models. And in our group, we work with both of these types of models, purely constraint-based or simulation-based models. But today I'm going to focus on symbolic semantic models, otherwise known as ontologies. And the great thing about ontologies, which I think is really fascinating. So ontologies are knowledge representation structures in the sort of most general sense, that you capture something that humans want to talk about, some way that humans want to talk about a knowledge domain in these structures, and they have basic principles of hierarchy, so you can capture your knowledge from the more general to the more specific. They have basic principles of interrelationships, and in general, a kind of logical language. You can use different logical languages, but the idea is that your logical language allows you to capture statements with which you can draw inferences in a structured and consistent and reproducible way. And ontologies, one of the great things about ontologies from my perspective is that they can be built by many people and serve as hubs around which a whole scientific community can put together its knowledge and its consensus, its sort of agreements on a topic. So for example, the gene ontology, which has been in development for more than 20 years, represents the functions and biological processes and cellular compartments, all of which are important for representing what genes and gene products do in biological systems, the whole sort of functional layer that we're trying so hard to interpret, but representing this in a consistent way and in a way that can be agreed on by different researchers, even if they're working on completely different biological problems, even if they're working on different model organisms, they're doing completely different types of experiments. And the power of this is that we end up with a whole lot of aggregated data which has used the same knowledge representation structure, so it amplifies a lot the sort of individual signals that each lab has produced in an experimental sense. So community consensus has power because it allows you to bring together much bigger data sets under the same umbrella. It also has power because it forces you to sort of get clearer. So each individual group might have their own perspective on what that particular area of biology looks like, or how it should be defined, or what are the interesting questions. But when you bring together lots of groups and force them to sort of negotiate a shared representation, you end up with something that at least is acceptable to everybody, and that improves and continuously improves the representational structure. So ontologies are typically developed by and for a community of researchers, and until recently have been mostly manually maintained. It's not easy work building an ontology, it's difficult work. And they're based on curation of knowledge from the scientific evidence base or the sort of experimental knowledge base. And as a result, modern ontologies are incomplete, and they represent decisions about relevance. What was important to the people who were generating the knowledge? What was important to the people who were curating the literature? What did they think were interesting parts of biology to include in the knowledge resources. So this is a limitation of knowledge resources, especially if they are manually maintained. They're necessarily incomplete and biased in the sense they're not, I mean, data-driven models are also biased by what data is available, but here we have active selection processes. What do we decide to put in? I'm giving a particularly special mention to CHEBI, which is an ontology of biologically interesting chemistry, which I happen to work on for a number of years and still an active contributor to. And CHEBI does present a classification both based on chemical structures or the features of chemical structures and based on biologically relevant chemical activities in biological systems. And it also includes information such as the molecular structure and so on. And it has a good collection of around 150,000 small molecules. So it's not anywhere near the same order of magnitude big as PubChem, which is 100 million. But it does cover most of sort of normal biological metabolism and drugs and interesting biologically active chemicals for various other reasons. So there are challenges with biological knowledge. The formatting of my slides is slightly different to what what I submitted. I hope it doesn't cause any problems down the line. Biological knowledge resources are hugely incomplete. They're far behind the sort of knowledge we have in the literature space, in the database space. They're biased by historical choices about relevance. They're slow and difficult to maintain and persistently underfunded. I mean, recently the NIH, who used to fund a lot of biological databases and knowledge resources, has been cutting funding for all of these things, leaving a lot of central biological resources which have been underpinning discovery in the biosciences for the last 20 years, currently saying, you know, help, we don't think we can continue anymore, which is really a difficult situation to be in. So the question that we're all, I think, somehow interested in to some extent, and various different parts of this overall puzzle are, can we have the best of both worlds? Does it make sense to think of this kind of cycle of knowledge generation and AI in that as a sort of AI in the loop in the broadest sense? Can we use our knowledge resources to improve our generative AI, to make it more relevant, more interpretable? In the areas where we need safety, can we use these knowledge resources to improve the safety and guardrails? And in areas where we need reproducibility, which I will say more about in a bit, can we use our knowledge resources to enforce or at least to measure reproducibility in a consistent way? And on the other hand, can we use the generative AI, this great technology which we have access to now, which is transforming all of these ways that we're working as scientists, can we use it to help scale up our knowledge resources, to improve the annotation of data with knowledge resources, which would improve the data available for training models in the future? And can we automate a whole lot of things that previously were just manual? So I want to talk about those two arrows, just some projects, some research areas which we have been doing in my group. And I think there's much, much more to talk about in this area than these one or two small examples. And I think it's a very exciting and kind of central aspect of where the field will be going in the next few years. So generative AI supporting knowledge resources. So as I mentioned, the manual growth of ontologies is painfully slow. There you see a kind of plot of the growth in terms of numbers of curated entries in CHEBI over the last 20 years. And this, not only is it slow, but it has been decreasing. This is because funding has been decreasing. So the growth of entries that are manually created is at best linearly related to the number of full-time curators that are employed, right? And that's the frustration of manual work. On the other hand, the number of molecules which we sort of want to have knowledge about in this structured way is several orders of magnitude bigger and also growing at a rate which would. So this kind of circle will never be squared. We have to do something differently. So the question is, can we use modern generative AI to automate the inclusion of new molecules into this knowledge resource in some kind of structured way? For example, we may have measurements or data sets of chemical structures, which we want to include in a chemical ontology. The question itself is, of course, broader. Can you apply ontologies automatically to newly measured DNA sequences or plain text experimental descriptions, etc., etc.? The general problem ontology application to some or other data type where that data type is appropriate for having that knowledge applied to it. And the structure of this problem is quite interesting because the ontology itself, as a hierarchy of classes, it's a hierarchical multi-label prediction problem. And it has a sort of interesting feature that what the human is interested in, what the human wants, is a more specific annotation. But for the more specific annotations, you naturally have fewer examples to learn from. So you have a much sort of poorer performance of an automated model. And whereas high up in the hierarchy, you have many, many examples because almost the whole ontology sort of converges around the higher level elements in the ontology, this general classification. This is easy to automate, but it is not very interesting for whoever wants to use this data. There isn't very much information content higher up in the hierarchy. So we have a kind of problem where it's not necessarily easy to do this automatically because what you want is not aligned with where the data is. And that makes it an interesting problem for a computational researcher. So chemical structures, of course, provide tons of classification relevant information. There is just some examples there in this illustration of different substructures within one given molecule, which may lead to different predicted classifications within the structure-based classification. And as a general principle, you can use different, again, architectures or problems, different architectures or models to try and solve this problem, right? You can represent your chemicals as a linear string and use transformer-based models. You can represent your molecules as graphs intrinsically and then use graph neural network-based approaches. You can use logic-based approaches, which are working, for example, through rules, which are identifying aspects of the molecule, which are very characteristic and rules-like, so you don't even need to use a data-driven learning system at all. You can even use software like deterministic approaches to do classifications. And our idea is that we would like to sort of explore all of these to see which works for which branches of the ontology. And in general, we have been following this research program for a number of years. So for this problem, I mentioned that it was a multi-label classification problem. For each molecule in the data set of existing already classified chemicals, we produce a label data set which corresponds to the structure of the ontology. For every class that chemical is a part of, we put the label data set to 1 or presence. For every class in the ontology that chemical, that particular input molecule is not a part of, we put in the label data set 0. So this labeling data set, clearly, in order to learn anything, it cannot be every class in the whole ontology because that would involve classes which only have 2 or 3 members, and there's no way to learn anything in a data-driven way for two or three members. And also, more importantly, there's no way to evaluate because we need some sort of ground truth, which is not part of the training data set in order to evaluate model performance. So we do use a kind of cutoff. We only include classes in this labeling data set if they have, in the past, we used at least 100 members. and now we use at least 50 members. And we have tried up to about 25 or 20, but we do see that these very smaller classes, it's very difficult for the model to learn anything useful in almost all cases. So at the moment, we're sort of fixed on 50, which gives us about 1,300 labels in our overall labeling vector. So the model is learning simultaneously 1,300 classes. What you see here are some typical performance profiles of some of our models. These are both, one of them was a sequence, they're both sequence-based models, but one of them was sort of prior to the transformers, and then our first transformer model, which certainly outperformed the prior sequence-based models. And I just include these because I want to mention some features of the performance of this problem in general. So on the left-hand side, yes, it's also left-hand side for you. On the left-hand side, you see a molecule-wise performance. So for each molecule, there are multiple classes predicted. So we sort of have a distribution of F1 scores of class performances for each molecule. And on the right-hand side, you see a class-wise F1 score distribution, which tells you per class in our 1,300 labels. I think back then it was probably the 900 labels of the 100 size thresholds. And what you see immediately is that both for molecules and for classes, some are performing very poorly. This is much more prominent for the molecules. Some molecules we cannot make any correct predictions on, right? That's this sort of peak at 0. So these are molecules where the model is saying, I don't like this molecule. It doesn't look like anything that I know anything about. I'm not predicting any of the labels to be true. And in fact, because we're using obviously a split on our input data set, those molecules were also part of KEBI, so there's nothing special about them. It's just that perhaps the classes that they're coming from were relatively small in the data set. So we really need, and we're constantly working on ways to boost the learning of smaller classes to avoid this kind of dropout on the molecule-wise performance. And on the other hand, you do see this distribution of performance on class level. This is correlated, but not solely driven with class size. So on average, we certainly see that smaller classes perform more poorly, but we also see that some classes are very learnable even if they're small, and some classes that are very big are nevertheless still having a poor performance. Okay, so we've put a whole lot of prediction systems together into this tool called KEBifier, because it takes input molecule and outputs KEBI classification. I'll skip quickly over the sort of more interface related aspects. If you want to try the tool, there's a QR code there, and the source code is all openly available as well. What I wanted to emphasize again is this idea that you have these different architectures, and in fact we have by now an ensemble which has both sort of transformer-based, sequence-based models, as well as graph neural network-based models, as well as logic-based models and procedural models, all in the same architecture. And this ensemble structure itself is quite an interesting thing, because we sort of need to have a sensible system for weighting the different predictions when the models don't agree, and this is something I will mention a bit more later. When the model can give an explanation or some sort of interpretability, we can show it. Not all models can give interpretability. We did for a while have the sort of attention weights of the transformer models shown directly to the users, and all our users were like, what is this? What do you expect me to do with this? So we're currently working on a more sophisticated way of bringing that information to the users, because they were not happy with the raw attention weights. Okay, and now I want to come to the next part, which is this idea that prior knowledge, and maybe I think this part might be more interesting for some of the people in the audience, this idea that we can use these knowledge resources to make better generative models. And I think this is really something which has great potential to really enhance, especially those of us who are working in lower data availability or higher complexity areas. So one of the challenges with generative AI, which we are particularly concerned with in my group, is the problem that although you may have good performance overall, your performance on individual tasks, such as annotations, may be not reproducible. The model may give different predictions on different runs. And this is kind of part of the technology. It's on purpose that it does this, but it's a challenge if you want to sort of rely on these annotations for any reason. It's a challenge where you are in some sense producing unreproducible science by using these models which do things in a potentially not reproducible way. And the scale of this problem is different in different areas. There was in the area of sort of structured annotation of research methods, which is an area where we work and where we apply natural language models, so sort of working on the research literature as the input data, published papers, and then you want as your output a kind of structured representation of what was done. And this plot is showing the reproducibility of the extracted structured representation. And if you only looked at the accuracy, this model would have had pretty good accuracy and does have pretty good accuracy. But the fact that sometimes it produces different structures for the same paper is something that concerns us. And it's difficult to envision how this problem can be solved because it's kind of part of the fundamental, let's say, nature of this technology, that it produces different things in different runs. That's one of its great advantages on the one hand, but on the other hand, we need to have reproducible pipelines for scientific research. And again, I want to remind that if you look at most data sets, the sort of general classes are overrepresented, but the specific ones, the sort of more detail level ones, they are less well represented. They're more likely to be not reproducible. They're more likely to be harder to automate. And they're also kind of hidden in average benchmarks. All right, so there are, of course, a multitude of strategies to improve reproducibility. You can vary your model parameters to sort of reduce this non-reproducibility, but that tends to impact performance and needs to be tuned for each problem that you're working on. And this is something, therefore, that in a way, if you take the same pipeline and apply it in a different problem, you may end up with different results. There are, of course, the possibility to give models, and this again is talking maybe more about natural language models, to give them more of the structure that you want into their context, and there are different strategies for doing that. But all of these approaches have their strengths and limitations, and at some point do not directly solve the problem of unreproducibility. And here you see just evidence that the performance, again, is kind of on average, especially on the right-hand side there, because what you're looking at there, the bubble size is how many examples were in the sort of data set for this particular class, and then what the performance in terms of precision was for the model predicting class. And in this, so you see that the more common the class is, the better the model is at figuring it out. But the more specific the class is, the more we're interested in it, right? So this is kind of more evidence of the inverse problem that we're trying to solve. So coming back to this KEBI prediction tool, we distinguish two different elements when we're putting model predictions together, having in mind that all model predictions may be wrong. Of course, we measure how well each individual model performs on a held out validation data set. And we call this performance trust. So for each class in the whole labeling data set, we measure that particular model's performance when predicting that class and say, we trust this model that much in making predictions on that class. And we also have a sort of internal rating from the model itself, which we call confidence. And this is the model's own estimate of how sure it was when making a certain prediction. And we put those together in the weighting where we sort of try and determine the overall when we have mixed inputs from different models. So this, I'll skip over this. The slide is slightly off. But basically the point is that you can use ontologies as a kind of strategy for doing a kind of after effect cleanup of your predictions. And this is okay. But the particular research stream that I wanted to end with is what we call ontology pre-training. So earlier on, I mentioned that we had been building models that were able to predict ontology classes. And at some point, we sort of asked ourselves, okay, if these models have learned this ontology, they've got the prior knowledge in them, maybe we could use these models for something else afterwards and see whether they benefited by learning this prior knowledge or not. Whether it helped them in downstream prediction tasks or not, whether it helped them, this knowledge from the ontology, this general knowledge from the ontology, whether it helped them to be able to make predictions from smaller data sets, for example. And in fact, we did see that this was the case. So our first experiment was with a chemical language model trained on the KEBI data set, given, first of all, some sort of general PubChem-based pre-training, and then given some specific training for, so after the general PubChem pre-training, we gave it KEBI training, so learn the ontology, and then we gave it a very small and very noisy toxicity data set. And what we observed was that after having had the KEBI training, the model was better able to learn from this very small and very noisy toxicity data set. And we were quite excited by that, and we think that there's something sort of general and potentially cross-domain, cross-purpose hiding in there. So we've been reproducing this in other data sets. We've been doing more experiments. Here you see, I think this was the, well, one of those data sets is illustrated on the right. And here you see on the left a table with several different data sets. And we've put this up to about 12 data sets in our latest study. And we also introduced a model which had been given a kind of sham ontology pre-training, which was we took the same data set in terms of the instances, the chemicals, as in KEBI, but we generated a fake ontology to go above it and sort of showed that this fake ontology didn't improve the model. So it was like a control condition. So this improvement that we see is kind of robust, and we're quite excited by it. And we think that there's something hiding there that maybe we could take our knowledge resources and use them to sort of preload our general purpose biological AI systems with this structured knowledge from human theoretical frameworks in a way that helps the models to learn more quickly and to generalize better beyond the training distribution on these small, noisy, and tricky data sets, which we typically in the end need to apply them to. That also doesn't show up very well, but we were able to show that this ontology-based pre-training also sort of increased the information content in the transformer attention weights by increasing the sort of level of meaningful subclusters within that attention weights. I mean, attention interpretation is itself a very active area of research, and one which has many potential challenges, but you can sort of see that there are more clusters at the bottom than at the top, but this, unfortunately, figure quality is not terribly great here. But yeah, again, sort of exciting that the model, after having learned the ontology, also had more sort of structure in its internal representation. Okay, so I want to conclude with some finishing thoughts. First of all, given that the typical biological knowledge encoded in ontologies or in other high density knowledge resources in biology are typically very challenging tasks for even our huge data-driven models to work with, because they are multi-label, hierarchical, very, very large numbers of classes to distinguish, for the model to distinguish, and relatively small numbers of examples for the model to learn from. And even when we pass like the whole ontology to context information or whatever, the sheer size of the number of distinctions that have to be made is something which typically results in poorer performance for our generative models. And this is exactly, I would say, sort of the same level of expertise that human experts have to have. They really have to become specialists in order to apply these ontologies. And specialists in a domain is something which is actually difficult still to produce in our generative AI paradigm. So another thing which we see is that the performance of such models can be different, and especially there are relationships between model architecture and performance on particular subparts of the problem. Not all classes, even if they have the same amount of support or the same number of examples to learn from, they don't all perform equally well. There's sort of semantic reasons for model performance. Some classes are simply easier to learn than others. And this is very interesting for those of us who work on methods. So there are a set of methods and strategies which we can follow and which are in development to sort of teach these detail level classification sort of ways of working to these generative models. And there are many open research questions in this area. How do we do this best? How do we take what we already have and purpose it for this? But we've seen that when we do this, or when we do it at least in some ways, this also has potential benefits for the generative models themselves, makes them better at understanding the domain that we want to apply them to. So with that, I say thank you, and I'm looking forward to your questions.
So, any questions?
Hello, hear me well? So basically, thank you. And I have some question. So basically, for NNN model in, you hear me?
Maybe that works.
So basically, if I understand correct, because I have done something in chemistry also, in applying AI in chemistry. So you said that for an NM model, apply in chemistry, for like there's a lot of product that not aligned with chemical property, if I understand correct.
That there are what?
There's a lot of product or the output of an NM model that not aligned with chemical property for the generative AI.
I think it would depend on what you were applying your model to do.
So for example, in SMI generation, for example, USPTO dataset. So basically, the observation that I read on some paper that said, okay, for a model such as a transformer-based model, they sometimes or a lot of time generate a lot of syntactically incorrect output. So what is the solution? Like I see that NNM can be constrained in the way that they must align with the chemical property. For example, if you generate certain product, the next thing that should be generated must be limited in the certain space.
Yeah. So this is so the question is, how do we make our models produce only biologically plausible outputs, right? Because they learn, they have to produce a number, and some of the numbers that they produce are completely biologically implausible, and we need to constrain them with knowledge to get them to do biologically plausible outputs only. Am I understanding correctly the question?
Yes, exactly.
Yeah.
So right now in the domain, they use the post-processing filter, or they use like directly interact with the logic of the model.
Yeah.
So
I think there would be different strategies of doing this. There are, I think this slide somehow got a bit lost, but it was part of my introduction. There are different strategies to bring the knowledge in to the model. And one of them is you could build like the architecture of your model in a custom way so that it can only produce those kind of outputs that you're interested in, right? And that means having a special type of last layer, which is only producing, but this may be quite problem specific, and maybe it's overkill for many problems. Then you can have, as you said, post-processing, which just throws away the junk and uses the model again, which is wasteful in a way. And something which we use is something like custom loss functions. So you put into your loss a heavy penalty for having a biologically implausible output. And then the model is forced to aggressively learn the sort of range that is more plausible because of this input of this penalty in the loss function. But this is also not panacea. I think there are custom solutions for different types of problem of this type where you would need to think a bit, but AlphaFold has quite complex representation of biologically plausible or allowable geometries, for example. And it needs this in order to produce biologically plausible structures.
I also have another question, so sorry about this. So basically, I have a question about ontology. So if I understand correct, ontology is something like the knowledge graph based off the chemistry or something like that, I believe so. So you said that you pre-train the model on ontology so that the model will know better about the domain thing, I would say so. But there's a paper in NNM that said that NNM model is not good at graph extraction information. So do you have any solution about this? Or?
I mean, I think it's not true that all architectures are equally bad at graphs, right? I mean, transformers are kind of a sequence-based. They're less good at the graph elements, but graph neural networks are designed specifically for graph representation learning, and you have representation learning strategies which try to learn faithfully what information is in a graph. But what we do is mainly the hierarchy. We mainly teaching the model about the hierarchy from generalized to more specific. And this is a little bit different to a graph problem. I think you could think of it as trying to learn trees.
Okay, any more questions? Yes.
Hi, thank you for a nice talk. I have a question about, maybe I missed it. How do you use, or do you use literature, generally bibliography in your learning your models? Because that's what Curato would be based on, right? General papers and softer phenotypes and things like that, which are harder to encode. So how do you use that?
So if I understood correctly, the question is, how do we use literature? And for the models that I represent, that I showed you that worked from a chemical structure, we're not using literature. We have normal, I guess, natural language models, which we use and apply to work on literature. But we are very interested in multimodal models, which have on the one hand, the chemical or another sequence information model, which is kind of fused with natural language literature processing model. And we're currently doing an experiment with different alignment strategies. And our hypothesis is that with a good semantic alignment, the fact that you have this layer which knows about the sequences or the structures of the actual data type would improve the natural language model's performance at answering natural language questions about those types of molecule. But I can't tell you whether the experiment works yet because it's currently just running.
Okay. One last quick question. All right. Then thank you very much for the amazing keynote.